afternoon, everyone. I'm Dr. Nermin Salah from the German University in Cairo, Egypt. It's a pleasure to be with you all today. I'm going to discuss a topic which is a little bit different from what you've been hearing since the morning, so I hope it's a little bit interesting for you all. I'm discussing part of research which is currently going on with my research group back home. It's regarding design and synthesis of novel non-CYP2D6 mediated tamoxifen analogues. Breast cancer is representing 25% of cancer cases in women. It has a mortality rate of 12%. One of the common adopted protocol for treatment of breast cancer is hormonal therapy. This is via estrogen receptor, estrogen synthesis antagonists like aromatase inhibitors, or via estrogen receptor antagonist, which is mainly through a class of selective estrogen receptor modulators, which is known as SERMs. SERMs is characterized by having a dual action. They have the ability to bind to estrogen receptors in some cells, like in breast cells, for example, where they do not cause any activation and they cause no breast cell proliferation. On the other hand, the same molecule can bind to estrogen receptor in other kind of cells, like uterine cells, and on the contrary, they cause activation and lead to more uterine cell proliferation. This dual action has been attributed to the differential ER alpha and ER beta expression. These are the two forms of estrogen receptor available in the body, or due to the different ratio of co-activator and co-repressor proteins in different cell types, or to the ability of estrogen receptor to take or to adopt a certain conformation when a certain drug binds to the protein. SERMs has been classified according to their chemical structure to different classes. Most famous, the triphenylethylene class, the benzopyrenes, the naphthalenes, the indoles, and the benzopyranes. Triphenylethylene is the class of interest in which my group is currently working on. Tamoxifen is the most famous example of triphenylethylene SERMs available in the market. In 1966, it was first investigated as an anti-fertility agent. In 1971, it underwent the first clinical study to show its effect in treatment of breast cancer. In 1973, it began to be marketed under the name of Nolvadex. In 1977, it was also approved for the treatment of breast cancer in postmenopausal women. In 1998, it was taken as prophylactic for women with high risk of developing breast cancer. It's not a surprise that at 2001, the sales was around $1 billion. Tamoxifen is sometimes perceived as a prodrug, which needs to be activated via CYP2D6 to the more active 4-hydroxytamoxifen, which is then further metabolized by CYP3A4 to the demethylated most active metabolite, which is indoxifen. This shows that CYP2D6 is one of the most important enzymes to form the active metabolites with a perfect clinical outcome. CYP2D6 pharmacogenomic studies proved that it is present in the body with different alleles, and these alleles led to the presence of different phenotypes, majorly poor metabolizer, intermediate metabolizer, extensive metabolizer, and ultra-rapid metabolizers. This led to the fact that not each patient are getting the same clinical outcome out of the disease. And so our research objective was trying to design and synthesize tamoxifen analogues which can overcome or bypass the CYP2D6 mechanism, and so we can ensure equal clinical outcome for all women treated by tamoxifen. We majorly did this, I'm not sure if this is working. We majorly did this via adding an ester group at position 4 so that we can avoid CYP2D6 and have an esterase working instead of this. We also worked on this basic chain, which is reported to have a strong effect on the anti-estrogenicity of the drugs. And we also have modified some structural modification, changing this ethyl group into a methyl group. To prepare these compounds, this is a little bit a chemistry part. We started with dihydroxybenzophenin, 4,4-dihydroxybenzophenin. Then this underwent what we call a McMurray coupling reaction with acetophenone to produce the triphenyl ethylene backbone. Triphenyl ethylene was then reacted with ethyl amino HCl salt in order to form the ether compound. This was finally esterified using acid chloride, appropriate acid chloride in a basic medium. All our compounds was tested for their ability to inhibit MCF7 breast cancer cell lines. They were also tested for the ability to bind to both estrogen receptor, ER alpha and ER beta. These are summary of some of our results. Compound 3, which have the hydroxy group, which is more similar to the hydroxy tamoxifen, have shown MCF7 less than 1 micromolar. It has shown an IC50 for ER alpha binding 8 nanomolar and 0.04 micromolar for AR beta. This gives a selectivity ratio of 45:1, which is more selective toward ER alpha. Compound 8, which is our ester congener, this is very similar to 3, but instead of the hydroxy group, we now have the acetate group. It showed an MCF7 of 2.6 micromolar and 0.007 ER alpha binding, and still it showed better ER alpha selectivity. Compound 10 with a decanoate ester have also shown an MCF7 of less than 1 micromolar. Compound 10 was further tested by the NCI over 60 cancer cell lines. Results was very promising, especially for MCF7, where we had an activity of gross inhibition, 50% gross inhibition of 5 nanomolar, which is 300 times more active than tamoxifen. The compound was also active over a panel of the 60 cell lines, which indicate a pan activity, and indicates also that this compound is probably working not only via ER mechanism, but probably through other mechanisms. We think it's probably mostly due to some apoptotic effect. To further validate our model, our collaborators in the United States have done a metabolite identification for the compound, where our compound has been incubated with human liver microsomal enzymes in presence of NADPH and in absence of NADPH. Our compound was metabolized to the active hydroxy metabolite in presence and in absence of NADPH, indicating that CYP2D6 is not really involved in formation of the hydroxy metabolite. We have also incubated our compound with 2D6 supersomes, and no hydroxy metabolite was present, indicating no role of 2D6 in the metabolism. We also incubated our compound with human hepatocytes, and we have seen the hydroxylated metabolite. Additionally, the glucurinated metabolite was formed. So this proves our approach, which indicate that we can really prepare some compounds which can avoid CYP2D6, and somehow this is more into the personalized medicine approach, which is currently adopted by many researchers. We did a molecular modeling to our compound, where we have overlaid compound 8, the most active compound, over hydroxytamoxifen. If you see, both compounds nearly take the same binding mode. One of the critical distances for binding is the distance between the cationic nitrogen of both compound and aspartate 351. In both cases, it was 3.6 angstrom and 3.8 angstrom, proving that we nearly have the same binding fit and the same anti-estrogenic binding mode in both cases. Also to see the 2D interactions of our compound, our esterified compound, compared to the hydroxytamoxifen, we see that our compound kept all the essential interaction, majorly the interaction with arginine 394 and glutamine 353, exactly like what hydroxytamoxifen do. Which means that replacing the OH by an ester group did not affect by any means the binding with ER alpha. Conclusions. We have prepared novel tamoxifen analogues showing an MCF7 growth inhibition of values between 0.25 micromolar and 3.97. This is all the compounds are more active than tamoxifen, which showed 4.4 micromolar. All compounds showed ER alpha binding affinity in the range between 7 and 200 nanomolar compared to tamoxifen. Compound 3, which is the hydroxylated compound, showed 10 times higher ER alpha binding, which again showed more ER alpha selectivity. This is very much preferred for treatment of breast cancer. Compound 10, our esterified compound, showed 300 times more growth inhibition, median growth inhibition when compared to tamoxifen. The esterified compounds were stable towards CYP2D6 and are rather metabolized via esterases. I need to acknowledge the people who were with us in this work. This is my research group, Professor Dr. Ashraf Abedi, Ms. Nihal, who had done the major part of this work, Ms. Rasha Rashid, Nuran Sawaf, Nurhan, Iman, Mr. Muhammad Saleh, Ms. Mirna Vector, Minna Hani, Tamim, Miriam Alber, Huda Alim. We have to thank Professor Dr. Rolf Hartmann and Dr. Matthias Angel for hosting some of our students at their lab in Zurland University. I have to thank the Science Technology Development Fund for giving me the grant to proceed in this work. I have to thank my mentor, Dr. Bianca Leader at Genentech, United States, for doing the metabolite ID. And finally, we have to thank the NCI for doing the cancer screening assays. Thank you for your time.
Any question for Dr. Nermin Ahmed?
I'm sorry, I want to go home too, but just one question. The rationale, tamoxifen is metabolized by CYP2D6. And that's the basis for have slow responders and ultra-fast responders because of pharmacogenomic effects. Exactly. Now you suggest, okay, let's chemically modify tamoxifen in order to avoid this. Yes. But why, how do you know that this modified tamoxifen is not being metabolized by other P450 enzymes, which then in turn can also have a different pharmacogenomic effects?
Yes, this is definitely possible. We even have claims that esterase itself has some allelic variation, but as a percentage, CYP2D6 allelic variation is reported to be among one of the highest variation available in CYP450 enzymes. So it's a nice approach, but we're just focusing on this CYP2D6